Hello everyone, my name is Enrique and this talk is about the assignment of T-cell markers to hematological and immune cell type based on single-cell proteo-transcriptomic data using machine learning approaches. First of all, I'm going to show you the workflow that we have followed to this analysis. But the most important thing that I have to say here is that we use our software and Seurat package for the whole single cell transcriptomic analysis. And the different steps that we did are the quality control, in which we delete genes and cells depending on the expression levels and the percentage of mitochondrial genes expression. Then we normalize and scale in the data. Then we perform a dimensionality reduction because in single cell RNA sequencing we have a huge amount of data, so we have to reduce all the dimensions. Then we did a clustering in which we separate the cells in different groups depending on the level of expression. And then for the different clusters that we obtained, we look for the most differentially expressed markers. Once you know the bioinformatic workflow that we have followed, it's really important to understand now the dataset that we work with. One important point here is that dataset 1 and 4 are supervised, so we know the cell type assigned by flow cytometry, and we employed this dataset to random forest analysis, that is a machine learning approach. And another important point or aspect is that we only used the fourth dataset in the last part of the analysis. And the interesting point of this dataset is that it's multimodal. So we have transcriptomic and proteomic data inside the same dataset. Now, the last thing that showing the results and the cells that we work with. Regarding the genes, we work with three genes lists. The first one is about cluster of differentiation markers, a list obtained from human protein atlas with 369 CDs, and they are really well known in the immune and blood field. Then we have list 2, in which we have all the genes given in the different datasets. The last list, list 3, that is composed of the 369 CD markers plus 63 membrane protein coding genes that we obtained because they were key in the isolation of different cell types and subtypes in these blood and immune datasets. Then we have the cells. We group them in the three different levels that you see because we evaluated the power of the different gene sets to identify these cell types and subtypes in the different levels. And the colors around the cell type show what the datasets in which these cell types or subtypes are present. Regarding the results, we can see here the clustering results with the three different gene lists and the first three datasets. The key point in this slide is that we always obtain the same major group classification. We have a group with B and plasmatic cells, another group with monocytes and progenitor cells, and the last group with T cells and natural killers. But our objective in this work is to evaluate the different gene sets, so how can we do it? What we did first was to evaluate the cell disposition and separation comparing our three gene leads. Are the cells grouping together inside the cluster? Are clusters separated between them? We answered these questions thanks to silhouette algorithm. With this algorithm, a very good cell group clustered will obtain 1 point as the huge qualification. So with silhouette algorithm, we compare the mean of the qualification of the different clusters, and we see with this that the third G list, the list with 3D markers plus the membrane protein coding genes, was the best one. The next point in the evaluation was how can these gene sets identify these cell types and subtypes? Besides our three gene sets, we included here in the random forest study one of the most famous gene signatures, Thibersort. To perform this random forest, we did a cross-validation running the algorithm 10 times, and we definitely obtained the mean of all accuracies of all runs. What we see in this result is that top 30 genes of each cluster from the list 2, all genes, and list 3, again CD markers plus the 63 membrane proteins coding genes, were the best ones once again. These results showed that the 63 genes added to the commonly known CDs markers had a very good result in the point of classification of hematological and immune cells. Another thing that we did to fix robust gene signature for immune and B cells was this trajectory analysis. We did it in dataset 1 again because it was supervised and we have intermediate state in maturation cells. We can see three different differentiation lines: monocytes, B and plasmatic cells, and finally T cells with natural killers. Following these three differentiation lines, we can see how markers change the expression along the specific maturation line, and this compartmentalization of markers allowed us to improve and finish the gene signatures based on single-cell transcriptomic data. This is a brief selection of the markers that we studied. Some of them are really well known, but some others are not too much. So, why did we do all this study? Why did we select these robust gene signatures? What we wanted to do was to give another point of view with single-cell RNA-seq data of the very well-known world of immune markers. In this way, the most relevant point is that we obtain the, what we obtain for T cells. We obtain a clustering based on the naive and memory phenotypes, let's say more functionally classification, but not based on CD4 and CD8 markers. We couldn't see differences in the expression of these well established markers, for example, and that's what we are going to see in the next slide. Here we can see the multimodal dataset 4. When we see RNA clustering, CD8 and CD4 T cells were together, without differences. However, protein clustering, that is the most usual way to classify these cell types, allowed us to clearly discern these two phenotypes. With this, we can see that RNA and protein information are not the same. So, what's the better way to classify or identify these cell types? That's the key point here. Apart from that, the rest of the results, images A and C, that we see here in this slide, were used just to corroborate the clustering results and the gene sets evaluation that we see in the first three datasets. So finally, I put this slide to clear up what we did in this walk. In the topic of hematological and immune cells, the markers to identify its different types and isolate them are very well established, and an example of this could be seen in CAR T cell or CAR natural killers. With this example, we could see too the importance and the relevance of this topic that is too huge. The information is just set in the clinical outlook, so we can see with this the real importance of this topic. What we only wanted to give is another point of view. Now that single-cell sequencing is completely set in the scientific paradigm, we wanted to study from the single-cell point of view more accurate and functional signatures that allowed us to work with these cells in an early point to its real behavior. So that's all. Thank you very much for hearing me. And if you have any doubts or comment, please write me without problem.
Thank you, Enrique.